All right, it's 12 o'clock here in Boston, and as people are still trickling in, I would like to welcome you all to today's Dana Farber Connect Science seminar. And it's really my distinct pleasure today to introduce Kelly Nguyen, who is joining us from the MRC LMB in Cambridge, where she is a program leader and I would really say a rising star in the structural biology division. Work in her lab focuses on the molecular mechanisms and really atomic details of how telomeres, the ends of our chromosomes, are protected in the machinery that is involved in telomere maintenance. This is really a long, I would say, a very long-standing problem in biomedical sciences, as we know that shortening of telomeres is involved not only in aging, but also in the progression of many disease, and proper maintenance is really critical. But it's only been recently that we start to get some molecular insights, and it's really her beautiful application of complex biochemical reconstitutions and cryo-EM imaging that is providing us now some details on the inner workings of key components of the telomere maintenance complex, such as the telomerase. Prior to joining the LMB, she pursued postdoctoral studies at UC Berkeley with Eva Nogales, and I think that's where really her interest in the ends of our chromosomes started. And prior to that, she was also at the LMB working back then with Kiyoshi Nagai, who, as many of you know, probably left us way too early. And she worked on the spliceosome complex and really made seminal contributions to our understanding of splicing. Her work has been awarded many prestigious awards over the last couple of years, and most recently the Suffrage Science Award and Early Career Award from the Biochemical Society of the United Kingdom. And it's really my great pleasure to have her here today, and I'm very much looking forward to her talk. I just need to add one more housekeeping item. If you have questions, please post them to the Q&A tool. So not the chat, the Q&A tool, and we'll read them at the end to make sure we get to all of the questions. Now, Kelly, stage is yours. Take it away.
Thank you very much, Eric. So first of all, I would like to thank Eric for this very kind invitation, and it's my great honor to be part of this wonderful series. I've listened to some of the previous talks, and so I'm really grateful that through the pandemic we can still connect. So today, as Eric mentioned, I'm gonna tell you about our work on this molecule here called human telomerase. And the work has actually lasted for the past 6 years. And since I was a postdoc with Kathy Collins and Eva Nogales at Berkeley, and now continuing in my own lab. So the two main things that I hope for you to take away from today's talk is to explain why telomerase is important and why really interested in studying its structure. And the second part is why cryo-electron microscopy was really instrumental in addressing this problem. So as Eric mentioned, my lab works on telomeres, which is the region located at the ends of the chromosomes, and it's very beautifully shown here in this metaphase spread. I'm going to change it into laser pointer. I'm sorry about that. So as you can see here, these condensed chromosomes labeled with dabbing blue, and these dots at the end of the chromosomes, these are telomeres. And the reason that one can visualize and label telomere specifically is that they have a very special property. So they consist of these G-rich repeats, and in humans, the GTTTAG, so just 6 nucleotides, and they repeat themselves hundreds and thousands of times, and they also have this 3' overhang. And this can range between 2 kb to 100 kb in mammals. So along this mammalian telomeric DNA, there's a complex called the Shelterin complex, and that was discovered by Titia de Lange at Rockefeller, which can specifically bind to this double-stranded DNA as well as a single-stranded overhang, and thereby providing protection for this very end of the chromosome. And so then the chromosome ends are not degraded or being mistaken a DNA break, because it looks like DNA break, but it's not targeted by DNA damage response pathway. However, this linear structure is actually problematic for the ribosome to replicate, because if you think about the leading strand synthesis, there's not much problem there, but the lagging strand synthesis, you can't lay an Okazaki fragment outside the reading frame. So as a result, this leads to this inherent problem called the N replication problem. And so therefore, every single cell division leads to a shortening of the telomere. So when the cell is young, you have plenty of telomeres, and over time it gets shortened until it's too short, and then they can no longer protect the chromosome. And this would trigger a DNA damage response at the telomeres, and therefore the chromosomes all fuse together. And as a result, these cells don't live very long. They undergo growth arrest, genomic instability, and cell death. And this inherent N replication problem is actually happening for most of somatic cells, and that's, in eukaryotes, that's most of what's in our body. But there is a subset of cells in the body that can escape this problem, and this is include cancers. About 90% of cancers or so, stem cells and germline cells. So what these subset of cells have in common is that they turn on this molecule called telomerase. And as you can already guess what this does, here you have a telomere shortening effect due to the N replication problem. And telomerase actually adds the telomere back to chromosome ends. So therefore, these cells can maintain a stable telomere length indefinitely. And this is a way for them to gain immortality. And often in the media, sometimes telomerase is dubbed as the immortality enzyme. So, how does it do this at a molecular level? So, in fact, telomerase is a reverse transcriptase, but it's not a common one. It's a very specialized one. So it has its own reverse transcriptase subunits called TERT. And what's special about it is it has an RNA component, and this is called HTR in human. So within this RNA, there's a region that we call the template region. And half of this can specifically recognize the 3' end of our DNA, as shown here. And the remaining half can be used to synthesize the exact GTTAG repeat. And what is also special about it is that it can do this multiple times with each binding event. And this property that I'll come back later on is called repeat addition processivity, or RAP. So now I just told you about these two main components that is required for the synthesis of telomere repeat, but that's not actually the whole story, because what we know is actually in the holoenzyme in the cell, in human cells, there are more than just these two components within telomerase. And to illustrate this point, I'm gonna just show you the secondary structure prediction of the RNA. You can see here, which we can divide into 3 components, as like we do in structural biology. So the RNA have this template pseudonode domain and a conserved regions 4 and 5. And these two have been shown to bind to the RT. But as you can see, there's the entire third domain called the HACA domain, and that is suggested to be bound to various factors to form what we call the HACA ribonucleic proteins. So what are these and why are they here? So these actually not only specific for telomerase, a very large family of RNP in both archaea and also in eukaryotes, because they are RNP, so they all have this RNA component. And in archaea, they have a simplified version of the RNA, which has only a single hairpin RNA structure. In eukaryotes, generally, many of them come in the form of small nucleolar RNA or small carbohydrate RNA, and they contain this double hairpin structure as shown here, and with the H and ACA boxes flank these hairpin. So that's where they also got these name from, because these boxes are very conserved motifs of RNA. So on each of these RNA hairpin, there are a heterotetramer of 4 proteins. So discarin, NOP10, and HP2 and GAL1. So for today's talk, you don't need to know or remember the names, but just keep in mind that there's 4 proteins bind to each hairpin. So here, where there's 2 hairpins, there should be 2 copies of these tetramer bound, one on each hairpin. So when we started this work in 2016, there were, there's also a protein called TCAP1 that bind to this loop here called cap box. So when we started this work in 2016, there's only structures that single hairpin, HACA, RMP, and generally what they do in the cell is that these snow in the sky and be converted uridine to pseudouridine using this pseudouridine pockets as a guide. And as for those who've got the vaccine, mRNA vaccine recently, these mRNA vaccine, all of these uridine residues actually converted to pseudouridine for stability. And in the cell, these actually are very important for these conversions in the ribosomal and spliceosomal RNA. So it's extremely important in the cell, not just telomerase pathway. And so what we have at the time we started doing this work is this single hairpin HACA structure. That's where I got the cartoon from here. And we don't have a double hairpin structure. We also don't know how TCAP1 associate with this complex. So with that in mind, back then in 2016, there's kind of a model, kind of a cartoon model of what's in telomerase, which we call the monomeric third HTR model. And you can see why I emphasized on this monomeric term, where in this model, we have one copy of the RT as shown here, one copy of telomerase RNA in black, and two copies of this HACA protein, one on each hairpin, and also TCAP1 bind to this hairpin here. So it's actually quite controversial at the time because there has been many other proposals of what actually in the human enzyme, and the reason for that was basically there were work that showed that the size of this molecule is between 500 to 600 kilodalton, and what we have is somewhere in between, so 550 kilodalton or so. And then you can see the story evolve towards the end of this talk. You find even more intriguing components of the whole enzyme. But there are many other versions proposed in this model. One is actually, people have proposed that it's a dimer of third and HTR. So none of these other proteins, but just two copies of these two components. And some studies only found discarin, which is one of the four proteins here. So therefore they propose only discarins present. And some suggestions also being whether there's two copies of this or just one, and how TCAP1 bind to the complex is also kind of, there's conflicting finding. One suggests they bind to the protein part, one suggests they bind to the RNA part. So with that in mind, when I joined the lab of Evan Nogales and Kathy Collins, we're trying to address these questions that become rather, it's taking us about 6 years or so to get here. But throughout this talk, I tried to address some of these key questions where we want to know what is actually in the human telomerase holoenzyme and how are these components assembled. And once we have an understanding about this, we would like to understand how it recognizes telomeric DNA substrate. And last but not least is that this enzyme is involved in a range of premature aging diseases, and we want to shed light on some of these disease mutations. So with that, we want to identify first what are the key challenges of studying telomerase and why, even just figuring out the composition has been difficult. And for those who studied telomerase before would know that it's actually the main challenges to studying this is a very rare enzyme. So it's absent in somatic cells. You take somatic cells, you lyse, just don't have any activity, detectable activity. So even in the most highly expressing cancer cells, work from Tom Jack and also Scott Cohen has quantified that you only have 50 to 250 molecules per highly expressing cancer cells. So with that number, and you kind of work out how much you need to get enough quantity for structure studies, it would come down to the order of hundreds of liters of human cell to get microgram quantity. So the question is, how do we make sufficient enzyme for structure studies when we don't even know its composition? So to do that, we employ this, I find a very powerful approach, which is called in vivo reconstitution approach, which is a semi-overexpression, semi-endogenous approach, where we only overexpress the limiting factors in 293 human cells. In this case, we overexpress the RT, and we can incorporate a tag here. We also overexpress the RNA component in these cells, and then we let the cell assemble the complexes, assuming that all the other components are more abundant. They come along from the endogenous sources, and then using a protocol to purify them in an unbiased way, which then hopefully would pull out the holoenzyme from the cell. And to do this, we divide the two-step purification, where we purify on the RNA. We have an oligo that can hybridize specifically to telomerase RNA, and then we elute with a competitor oligo, and then we use a tag on the RT, which can put out on a second step, and then that allows us to obtain the holoenzyme. And also, because this is quite versatile, we can incorporate DNA substrate of our choice on this step here to get a substrate bound telomerase RNP. So having done this, we are able to obtain, we grow a lot of cells and transfect a lot of these in order to get to them, to the microgram quantity that we need. And as you can see here is a silver stained SDS page of the elution fraction. And you can see here all these proteins were identified by mass spec. And because it's an enzyme, so you can assay for its activity. So it's a polymerase. So what we do is we give it telomeric DNA and also nucleotides. And one of them is radioactively labeled. And if it's active, you can see a ladder of these products. And this is exactly what we see for this purify enzyme. We see a ladder of 6 nucleotide repeats corresponding to the telomere repeats. So now the next question is, having this enzyme, how do we determine the structure? So now in this scale here, I'm showing how different techniques that we use to visualize different biological objects. And visualization of biological specimen has been very powerful for centuries for studying, the object of interest. So ranging from atoms all the way to things that are visible by human eyes. So the range that we're interested in is this macromolecule range, where generally there are 3 main techniques that people often use to studying structures of these molecules, and that include X-ray crystallography, NMR, and electron microscopy. So knowing this, we had to choose which one is most suitable, and for various reasons, cryo-EM has been instrumental in addressing this question. And one of the main kind of advantages of cryo-EM over the other 2 techniques is that we can work with a very small quantity, and this includes this microgram quantity of sample that we can produce. And this is an order of magnitude less than the other 2 techniques. The other 2 techniques, you need milligrams quantity. And the second really powerful thing about this technique is that the bigger the molecule generally often is the better. And for the other 2 techniques, it's more difficult to study when molecules are large, or by NMR, for crystallization, it's harder to crystallize them. And last but not least, and we find that this is very powerful for us when working with the flexible complexes, that in cryo-EM, we can analyze a lot of these using in silico image analysis to dissect some of the composition and conformational heterogeneity. So with this information, I'm going to give a little kind of introduction of what the technique involves, and then I'm going to show you the data that we have later on. So like all the techniques, EM first also require a good sample that's biochemically well characterized. And then we apply it on this very tiny grid, which is about 3 millimeters across, as shown here. And then if you can see the side view of the grid, you apply about 3 to 4 microliter of sample. And then what we do is we plot most of it away, leaving a very thin layer of the sample on the grid. And then this is then get plunged very quickly into a cryogen, in this case liquid ethane. And the trick is that you freeze it so quickly that the the ice doesn't have time to form. And then what happens is your particles get embedded into this vitrified or glassy state of ice. It looks a little bit by looking out the glass window, and you see these particles embedded on it. And these are the particles that we subjected on an electron beam and then collect the micrographs resulting from this. So the second part of EM is image processing, which I mentioned. So we start with these electron micrographs when you have these particles and these noisy images. And what we do, pick these particles out, and then because they're noisy, so we, in order to get better signal to noise, we group them, the similar particles together, and basically average them to get better signal to noise in this step called two-dimensional classification. And another very powerful technique that EM can offer is you can do this in three-dimensional. So you can then sort your particle in different three-dimensional states, either compositional or conformational, and then choose the state of interest in order to obtain a 3D structure from this particular class. So we apply similar concepts in order to solve the structure of telomerase. And more recently, actually, EM is actually not a new technique. It's been around for decades, but there has been very important developments in detectors and also image processing. And that allowed us in about 2013, 8 years ago now, so that we can obtain 3 to 4 angstrom resolution for membrane proteins and also for the ribosome. And very recently, our colleagues at the OMB were able to obtain really near atomic resolution for apoferritin, as you can see here. And I would like to also highlight that it has been very instrumental in many of the SARS-CoV-2 structures in the last 2.5 years or so. So with that, we pretty much apply the same concept for telomerase. So we collect a lot of these electron micrographs, we put on the grid, collect these electron micrographs, get these different two-dimensional class averages when you see the particles sit on the grid in different views. And from this first side, you can see that they form a very elongated structure. So, and roughly, it can kind of divide it into 2 lobes, a top lobe and the bottom lobe, and they're quite flexible relative to one another. So in order to kind of get the best out of each lobe, we have to kind of work with them individually in image processing. So we start with a lot of micrographs at the time, so 12,000 micrographs. We picked about 1 million particles, and by this computational sorting that I told you, we're able to resolve the complex first to 8 angstrom or so. And as I 2 slides ago, I showed this map of 1 point something angstrom. This is pretty low resolution, but at the time that was sufficient for us to address some of the questions that we were interested in. So here is the reconstruction. So I'm going to give you a little movie of what we learned from this, and then I'm going to move on to our later work recently. So here is the reconstruction at 8 angstrom. At this resolution, you kind of see secondary structures of RNA and protein, and we can fit in 10 protein subunits in this map, and also telomerase RNA in fragments. And one of the things about the structures, we can see, I told you that there's 2 lobes, and from the structure, we can see that the whole structure is held together by a single RNA, as shown in blue here. And at the bottom of the structure, there's what we call the catalytic core, because the reverse transcriptase is located here. We also have a substrate in the active site, as shown here. And this is just rigid body dock. And on the top lobe, what we call the HACA lobe, and here we can fit 2 copies of the HACA tetramer. So this is the first one, second one, and then there's the WD40 domain at the top of the structure belonging to TCAP1. So by fitting these models, we're able to have a little an architecture of the enzyme. So straight away, it really supports the monomeric model of the enzyme. Because we can fit 1 RNA, 10 proteins, and among these 10 proteins, only 1 copy of the RT and 2 copies of these HACA proteins and TCAP1. And the structure had these 2 lobes, and each of them actually perform very different roles regarding telomerase actions. So the HACA lobe that consists of these 2 copies of these HACA proteins in TCAP1 is important for its biogenesis and also its localization to the Cajal body, the nucleus. Whereas the catalytic core has the RT, and this is important obviously for DNA synthesis and also recruitment to telomeres. So at the time, it's very revealing because we kind of have a rough picture of what the enzyme has. But as you can see that this composition, although it confirmed the monomer model, it's only temporary. So by the end of this talk, you see that this increases by 2 because we found still new subunits. 100% of this structure was modeled with homology models and RNA fragments. So this is also before AlphaFold time. So homology model is not as good back then. And so we do need the high resolution structure in order to understand how it works at a molecular level. So that coincided with me moving back to Cambridge in 2019 and set up the lab. And I'm extremely grateful to have those 6 months before we had to go into lockdown to pretty much get things up and running. And we're able to purify telomerase pretty much the same way. And it's very active, even though it's moved across the Atlantic. So with that, we had to ask ourselves, how do we improve the resolution? In fact, when I was with Evo, I tried quite a lot of different techniques to do this, but in the end, thanks to the OMB, which has a lot of computational resources, we decided just to brute force with the new technology and see how it goes. And that's how we actually solve this problem. And again, just to remind you the structure of these 2 lobes. And so how much we had to brute force. So back in 2017, that's when we collected data. We collected on the Berkeley Creos with a K2 detector. And since then, this has been upgraded to a K3 detector, which is not only faster, but also bigger. So you can collect more data in a shorter amount of time. And so back then we collect 12,000 micrographs, and this is over a week of data collection. And since then, we now collected for this data set 44,000 micrographs. And because it's bigger and faster, so we actually collected 15 million particles instead of 1. So the number of particles actually increased by 15 times. And also then we have, the image processing has also been improved then. So we have a new version of image processing software, and with the computer cluster, we can handle this data and process it through the pandemic actually. So that's how we make the big jump from 7 angstrom to below 4 angstrom. So what I'm going to show you now is kind of a movie, a teaser to kind of walk you through what we learn in this new structure, and then I'm going to go into details of the points. So this is also a collaboration between my old boss and also Ritu Das and his lab for helping us with modeling part of the RNA using Rosetta. So to start with, I'm going to show you the old structure again at 8 angstrom, and that includes these 2 lobes. And then I'm going to morph it into the new structure so that hopefully you can see the improvement in the resolution that we gained. And this we're able to see segment these individual subunits, and we have sidechain information, and that really allows us to build a full atomic model for this. So let's have a look at the catalytic core. So here we have the RT, and now we have a model for the RT and also the RNA, and the DNA substrate is in the active site. And this is, we build a full atomic model for this, and we actually show how the DNA interact in the active site of telomerase. And up here, for those who have sharp eyes would have noticed there's extra domains or extra proteins here. And these at the point with mysterious factors, and they're in fact histones. I'm going to tell you how we identify these later on. So up here we have the HACA protein complex, where we have 9 proteins and telomerase RNA. And here, because actually it's involving other processes, we also built a whole atomic model for this complex. And really, it's just belonging to telomerase, but it's actually had big implications. We have a full reconstruction when we have 2 lobes together because they're more flexible, so it's not as good. So we use Rosetta with Richard Das to build a full atomic model of the RNA, and that allows us to get this picture of telomerase with 12 subunits and the RNA. So now I'm going to go into details of these points that I highlight in this movie, and hopefully we'll walk you through these findings. So first, the first big question is, how does it interact with telomeric DNA substrate? So to show you this, I'm going to segment the catalytic core, where you have these third protein bind to these two domains of the RNA. And the RT itself has 4 domains. It has an N-terminal domain, an RNA binding domain, a reverse transcriptase domain, and a C-terminal extension. So these 4 domains kind of get enclosed by the RNA. So the RNA kind of form a cage around these RT domains. And in turn, the RT domain encloses the DNA substrate bound to the template, as you can see here in this cartoon. So it's kind of closes in on this duplex. The question is, how does it look in here? So let's zoom in and have a look what we got here. So to get this structure, we actually in purpose uses DNA substrate that has a TTA-GGG permutation of the telomeric DNA, and because it's end with GGG, so it binds most stably and allow us to get the structure more easily. So, and in the EM density, we observe exactly 6 nucleotide of this repeat, so TTA-GGG, and that nicely interact with the template in the active site at the bottom here. And actually, at first, when we use this, we assumed that there would be 6 base pair, because this TT we thought would base pair with this AA. But in fact, when we solved the structure, we were surprised to see actually only 4 base pairs formed. It's actually shorter than we expected, and the remaining 2T kind of split away, and so that's kind of surprising. And the question is, what was captured here? So as I promised earlier, I'm going to kind of explain a little bit about this property of telomerase called repeat addition processivity to understand what we capture here. So again, here is the substrate we used, and we just turn it around so that the 3' end of the DNA is on the right-hand side. So in fact, the end of the telomeres actually end with a different permutation from this. It actually ends with GGTTAG. And in order to initiate synthesis, telomerase will initiate on this GGTTAG end of the telomeric DNA. And it is template part of the RNA can be divided into 2 parts, the alignment part and the template part. So the alignment part will hybridize this 3' end of the DNA. And then the template part will be used by the RT to synthesize the 6 nucleotide repeat. And it does this consecutively in this elongation step. So you add GGTTAG. When it reaches the end, it has to terminate. So 2 things can happen. So this can dissociate, the DNA fall, or it can realign for another round of repeat synthesis. So this is called repeat addition processivity. Because we use this TTA-GGG permutation, we actually capture the reaction at this stage of the cycle. So we are capturing the complex at the elongation phase of this cycle. And of course, in the future, we hope to build this up and start capturing it initiation or termination in order to understand how this active site actually can cycle through this repeat addition processivity cycle. So that is one aspect. And the second aspect is, how does it add the nucleotide in with each nucleotide addition? So telomerase is actually very similar to many polymerases. It uses the similar nucleotide addition mechanism. So here you have a substrate and a template, and the incoming nucleotide is supposed to come. So it uses a catalytic triad that has 3 aspartic acids, which will catalyze this nucleophilic attack of this OH group to the incoming nucleotide and then that releases a diphosphate, and this will get incorporated into the growing gene. So here is the state that we often call the pre-nucleotide state. And here's after it's added the nucleotides called post-catalytic states. So we look at the active site. We actually, at the time, we actually didn't think about it. We didn't add any nucleotides or non-hydrolyzable analog. So what we have is actually an empty, not surprisingly, an empty active site. And we have, this is the catalytic triad, and this is vacant. And because it's quite you know, this opposite template residue is kind of ready to base pair. So because of this, we can model in an incoming nucleotide using this as a base pair restraint. So we can model in this DTTP from another structure. And what is striking about it is that this tyrosine that's really nearby and kind of stacked on this sugar here. And if you look at the C2 carbon on this sugar, if we put an OH group here for an RNTP, this will clash with this tyrosine. So it seems that this active site is already selected for a DNTP instead of RNTP. So it's still very cool to see. It's just a model, but from this we can see that this is likely to be that selection. So, and next is about this mysterious density in the core that I mentioned earlier and how we identify them. So in fact, when we published the 8 angstrom map, we have this unmodeled density that we did not know what it was, but quite a big bulk of it. So we kind of keep it in mind. So then when we have a new map, which is better resolution, we then ask ourselves, can we find out what's in this density? So we took kind of an unbiased approach. So we use the old crystallography trick called molecular placement. And this is developed by this pipeline actually developed by Alan Brown, who's at Harvard at the moment. And so what we did, we take this density and we ask, we search for a large library of PDB and see what fits in this density. It's very unbiased. So we tried first to fit things that interact with telomerase, but none of them really worked. So using this approach, to our surprise, the top hit that returned is actually histone H2B from this PDB code. And as you can see here, that's histone H2B in this mysterious density, but it only fit half of the map. So what naturally we take histone H2A from the same structure and fit it in, so it accounted for the entire density. So with that, we have an updated view of the telomerase catalytic core now with a histone dimer binding to telomerase RNA as shown here. So the time we actually found this was we were in lockdown, and we were very puzzled. How can we be sure, and what could they be doing in telomerase? So first we resend the sample from the mass spec, and we obtain a lot of isoforms H2A, H2B in the mass spec, but it's kind of, yeah. Initially, when we had in the mass spec, in my first paper, we actually say that it could be contaminants, and then we didn't actually include in the table. So what is the histone doing? So what we found is that the histone is actually binding to an essential RNA motif, which is this part called the conserved regions 4 and 5. And as you can see here, this form a 3-way junction, and here the histone dimer. It doesn't bind this alone, actually bind to this part of the RNA together with the RT domains. So this is the 4 domains of the RT, and that these are the TRPD and CTE that bind to this 3-way junction together with the histone. And what is most remarkable is together they actually shape this hairpin here that has been shown by many previous studies to be essential for catalytic activity of telomerase. So if this is not properly folded or mutated, telomerase is dead. So it seems to us that histone is taking part in shaping this very important hairpin in telomerase RNA. And another thing we analyze is how does it bind? And by looking at the surface, it uses a very positively charged surface to bind the RNA. And remarkably, putting this side by side with the nucleosome structure you can see here, it uses pretty much just very similar surface to bind the nucleosomal DNA and as well the telomerase RNA. So, it's kind of remarkable. It's curved, the nucleic acid backbone. And, my postdoc was still not quite happy with it. So what we did is that, can they bind in vitro? So he then purify human H3A H2B and also purify this fragment of the RNA and then gel ship them. And in fact, he did see a shift. So they did bind using this assay. And although the affinity of the H3A H2B to this RNA is actually lower than to nucleosomal DNA. So it's in the order of the specific binding is in the order of 35 nanomolar. So that's kind of cool. And the next question we also want to ask is, can this also accommodate H3H4? So is the binding site for H3H4 accessible in our telomerase H2A H2B? And what we did was we took a nucleosome structure from the same BTB, we took away all the DNA and we superposed that the octamer on our structure. And there is enough space to fit H3H4, although we don't see any density that would correspond to this, but this could in theory be there. So that really raised a lot of questions, more than we actually could answer. So there's anything more than just RNA folding. So first, given the density we see, we do not see the tails, for example, so we cannot know whether there's any variance, it's any histone variants or canonical histones. And also from the role of binding to RNA, we're now trying to investigate what its role is, because we see it binding, but we don't know what is it doing in telomerase. And we actually mutated some of these interacting residues, and it does affect telomerase activities. And this is to be reviewed in the future. And also another general question is, besides telomerase RNA, do histones interact with any other RNA generally? So that's also a big question one has to start looking at as well, because they might have a big implication in RNA biology if that's the case, or this is more of a telomerase specific example. So that's taking me to the last but not least part of the talk, is trying to understand the disease mechanisms from the structure. So in the telomere field, there is a very famous class of disease mutation called dyskeratosis congenita. And these many of these mutations were originally found in the HACA RNP. And in 1999, my boss actually found that these mutations actually resulted from telomere maintenance defects. So these patients that have these mutations suffer from bone marrow failure, premature aging, and they generally have very short telomeres, and they have defects in telomere maintenance. So the question is that, and for a long time, it's very difficult to explain many of these mutations, because from what is known structurally, it doesn't make sense. So by solving the structure, we're able to start having an atomic model for this part of telomerase RNP. So just to remind you again what these HACA RNPs are. So in general, they've come in the form of small nucleolar RNPs or small Cauchy RNPs. And these are very well known as ribosomal and spliceosomal biogenesis factors. And they have this double hairpin structure. And on each of this hairpin, you have this heterotetramer. What is not known is how these two hairpin come together. And by solving the structure, we can see how these two hairpin assemble these two copies of the tetramer. So I'm going to walk you through the structure. So here is the 5' hairpin. This is this one, and then this is the 3' hairpin. So the first tetramer binds in the 3' hairpin like this. The second tetramer binds along the 3' hairpin. And then you have TCAP1 cap, the top of the structure. So what we didn't kind of expect is that these two are not separated. They actually form very, very tight interactions. So these two tetramers have this very extensive interaction at the interface. So they're not just flexibly tethered by the RNA. And what is most remarkable is that when we map the disease mutations on this, that's where most of the disease mutations cluster. You can see here, I'm going back, and you put it on, that's this, intertetramer interface is pretty much the disease hotspot. So, of course, you can zoom in and have a look at this very, very extensive interaction between these carrier molecules. So obviously, these interactions would affect telomerase assembly, because if you disrupt these interactions, the top part of telomerase cannot form, and this is important for its biogenesis. So telomerase assembly is affected. So therefore, that's explain why these patients suffer from telomerase deficiency. So with that, I would like to conclude. So here, what I'm just show you is that how we use cryo-EM to really tackle this telomerase structure and really settle a long-standing controversy regarding what's in this enzyme. And the latest structure we have really reviewed insights into this active site. And of course, what we capture is only one state. So hopefully in the future, we're able to capture a full picture of the same active site going through this catalytic cycle. And also at the same time, we discovered a histone dimer as novel telomerase factor. And now we're currently investigating what their role in telomerase biology is. And last is we characterize a lot of these disease mutations that previously we couldn't explain why they cause telomerase deficiency. And so with that, I would like to thank many people. So I would like to thank my bosses at Berkeley for being extremely supportive. It was one of them, it was a really fun time working with them, and I had a wonderful 3 years in Berkeley. And our collaborators at Stanford, and also colleague Kapau, who's now at Broad, and all the new neighbors and facility, core facility staff and colleagues at the OMB for support, especially the last 2 years when you can't really talk to people very much, freely. And here are my lab and all these very wonderful people, especially George Garnim and Marika for doing the work that I just presented to you at the end. And Zala and Sebastian are newer members, and hopefully in the future I'll be able to tell you more about their work. Thank you for your attention as well.
Thank you, Kelly. That was wonderful. But we give people some time to type their questions. I'm going to start with something. So one thing that keeps puzzling me, and I don't know if you thought about that or if there's much known, how the histones get there, because free histones are extremely toxic to cells. There's very little of them that are floating around. Cells sweep them away typically quite effectively.
Yeah. So it's quite cool. So basically, when I didn't show this piece of data here, but I have a slide for that. So when we take telomerase and we capture them via histone antibodies, we're actually not every telomerase molecule has histones. So that's the nice thing about cryo-EM. You can sort all of this out. So actually, what we have is only a fraction of the complex have histones, and there's a few possibilities why that's the case. One, it could be some cell cycle regulated, telomerase population. Second, could be some sub-localization of telomerase in the nucleus. So currently we don't know exactly what this is, but if it's cell cycle related, it's actually it might not be a coincidence, but telomerase is actually most of the time it's not at the telomeres, and it's recruited specifically at the end of the S phase. And this is also when histones are most abundant, because it's after replication. So it could be a coincidence or could be not, but thinking about it, it might be something related to that. It could be a cell cycle or a subpopulation of telomerase in the nucleus that carries them.
Yeah. And directly following to that, so if I understand correctly, your sample that you purify is substoichiometric for histones.
Yeah, yeah.
So have you tried to just titrate in more free histones and see if it changes activity, if it stimulates or inhibits?
Yeah, we actually are trying to do this in vitro. We're actually trying to reconstitute minimal telomerase in vitro and titrate in. But the problem with that is that for our RMP to be happy, to have to keep it low salt, and the histones, when we purify them, they had to be, unless they bound to nucleic acid, had to be kept at really high salt. So that's one of the technical challenge that when I set my student to do, we didn't, until we done the experiment, we just ended up precipitating the histones in our experimental conditions. So now we're trying to first form a complex with the RNA first, and then bring the salt down, and then try to assemble that with the RT in this in vitro system and see. But yeah, so far that has been quite challenging. Yeah.
Okay, I have a question here from the Q&A from Peter Lansdorp. Beautiful work. Does the structure inform possible differences in repeat addition processivity between species?
That's a very good question. So currently not this one. So yes, it's a very good question. So in our structure, we only see 6 nucleotides, and in the one that we have but currently not published, we see a few more nucleotides, and that reaches to the point when we can see species difference. So this one not, but maybe give us a few months and we are able to give some insight in this, because yeast, for example, it has very low RAP, and we, potentially there's a motif that we found that it could be mostly conserving vertebrates, but it's not. So that could explain why vertebrate telomerase is more persistent than yeast. But yeah, that's kind of in the work at the moment. Yeah.
One just technical, as you said, you've brute forced your way in, and you collect that large amounts of data. Have you thought about brute forcing using a non-trap version where you don't use your slightly altered sequence, but you just try to start the reaction and collect data?
Yes, that's a very good question. So one of the problems, so one of the very old studies that showed actually telomerase is quite strange, because it has very different K on and off for each of the permutation in the catalytic cycle. So the one that we chose has, kind of the slowest K off. So if we do it in bulk, our guess that we will capture that state, whereas the other one has high K off, so we just follow up. So now we're trying to use some biochemical tricks to capture these different states. It's a bit harder to do, Yeah,
okay. Let me see if there are more questions. If not, people seem to be still tired from the long weekend, so I think
Martin Luther King's Day.
We'll conclude here. It was a great pleasure to have you, Kelly.
Thank you very much.
I hope I see you in May here in person.
Thank you very much for the invitation.
Thank you. Bye.
Bye-bye.